right?
Yeah.
While we are waiting, may I say a word or two?
Yes, please.
First off, good morning, everyone. I appreciate all of you. It's just a joy to be give great work presenting. And two things. One, I want students to acknowledge their professors, the advisor, committee members, and whoever else they want to acknowledge. I want them to acknowledge the funding mechanism for the stipend and for the research, where the funding has come from. So we need you to have the intention of asking questions that will inspire us all to learn from and be very respectful to our students. They are stressful enough with the presentation and all those things. So let us be gentle and maybe ask questions with the intent to inspire us to learn from each other. How about that?
Great.
Thank you.
Okay, good morning, everyone. Thanks for coming. Today is our 1, 2, 3, 4th presenters in this year of the graduate of NRES program. And today is Kusalika. She's a second cohort of this NRES program. And like a story from any other student, how did he came here? She was working with the Dr. Nimal Abesinga, who was here in 1 year sabbatical, just left in October. We are working together like past 3-4 years, and Dr. Abesinga was her advisor in Sri Lanka. So that we get connected, and we already had even some publications before she landed for joined the master program here. So that she's here. And she's also, after graduating, she's a very busy. She already get offer for the PhD program in University of Saxon in Canada. I hope I pronounced correctly. So she's just going from here to Sri Lanka and coming back to Canada to join for a PhD program. So today she's going to present about this the 3 different variety of sorghum that we grow, developed by Texas A&M, which we have collaboration in CBG funded project. So all is yours now, yourself. Let's welcome Ms. Kusalika.
Thank you, Professor. And good morning, everyone, and welcome you all to my thesis presentation. And thank you very much for coming. Today, I'll be presenting my thesis. And my name is Kusalika Kularathna. I'm a graduate student in Natural Resources and Environmental Science in Prairie View University. My research title is Carbon Sequestration and Greenhouse Gas Emission Dynamics of Forest Sorghum Cultivars in Southern Texas. And this research was conducted under the supervision of Dr. Ramre. He is my chair of committee, and other committee members are Dr. Gabrikadon Tefera and Dr. Richard Griffin. So let's move to the outline of my presentation. At first, I'm going to give a brief introduction about myself, and then I'm going to the chapter 1, its introduction. In there, I'm going to talk about the background of the research, and the statement of problem, objectives of the research, research questions, significance of the research. And then chapter 2, it's about the literature review and conceptual framework. And the chapter 3 is about methodology, followed by study side, research design, method of data collection, and statistical analysis. Then I move to the chapter 4. It is research and discussion. So I have listed the soil properties, plant biomass, plant carbon storage, total below ground carbon storage, total system carbon storage, and total soil carbon dioxide emission and methane emission. And then I'm going to conclude this presentation with few recommendations and explaining about my few accomplishments during these 2 years of stay. So let me briefly introduce about myself. My home country is Sri Lanka. It is a very beautiful island which is situated in South Asia. Actually, I was born and raised in the Kandy area. That's the middle of our country, and it is belong to the wet zone of Sri Lanka. But for my bachelor's degree, I had to go to the dry zone. It is in Anuradhapura. So my university is Rajarata University of Sri Lanka. So after graduation, I worked 2 years as a research assistant in two reputed universities in Sri Lanka, the Sabaragama University of Sri Lanka and the Peradeniya University of Sri Lanka. Actually, during that stay, I increased my knowledge related to soil science, and that time period encouraged me to apply for the for the higher studies. So here I am. So let's move to the chapter 1, introduction. Background of the research. As we all know, increasing greenhouse gas emission is one of the major threats that we are facing today. According to this, Jonas et al. 2024 claimed that this increasing pattern is directly impact for the climate change. And you can see there is an exponential growth in this greenhouse gas emission too. So when we talk about the agriculture is contributes 11 to 12% of greenhouse gas emission to the worldwide. And when we talk about the forestry and land use, it is 24%. That means the proportion is much high. So what are the sources for this greenhouse gas emission? So I would say the soil respiration and fertilizer usage, extensive livestock usage, as well as the decomposition of organic carbon matter. And in this case, this agriculture can play a major role here, as cited by Lawrence and Lal 2014. Our soil can act as a carbon sink if we manage it properly. So that's why this topic is very much popular these days. So in my research, I'm going to talk about the carbon sequestration. So what is this carbon sequestration mean? Carbon sequestration mean capturing atmospheric carbon dioxide and fixing through plant bodies and soil as organic carbon content. And different factors are affecting for this carbon sequestration, such as the root biomass, the root structure, and the amount of biomass the plant is going to produce, as well as the perennial or the annual variety, and the soil properties are going to directly influence this carbon sequestration. And the crops with extensive root network, as well as the crops produce much higher biomass, and some of the perennial, most of the perennial plants can contribute to more to this carbon sequestration process, since it is stay a longer period in this soil. So my research study is situated in southern Texas. For we all know, southern Texas has high temperature and the sporadic rainfall pattern as well. As this southern Texas is well known for the ranching industry. So the farmers continuously need forage crops to feed their animal. So there's a high demand for this forage crop. So that's why we selected forage sorghum as our research study. So when we talk about this forage sorghum, it only need a very limited irrigation and limited nutrient inputs, as well as this forage sorghum shows a high efficiency in water usage, as well as it is very heat tolerant. Also, it produces more biomass. And another thing is, this sorghum is a C4 plant. So that means it has a efficient photosynthetic pathway than C3, because the PEP is the one who receives carbon dioxide in C4. So because of that, the photorespiration process is not going to happen here. So there is no wastage of carbon like C3. So this C4 plant can fix carbon dioxide more efficiently than C3. So let's move to my research. So in my research, I have used 3 forage sorghum varieties, 1 annual variety and 2 perennial varieties. To your left side, you can see the annual variety. This is the annual variety. It's a commercial variety and the name is Sugargrass Ultra. And the middle picture, you can see the perennial variety. The name is PSH20TX01. And to your right side, you can see that second perennial variety. It is PSH09TX15. And in these slides, I'll briefly explain about my forage sorghum variety. So the first one is Sugargrass Ultra. As I mentioned before, this is an annual variety and it is a high yielding variety too. So you can see the maximum growth is above 3 meters. So you can see in this picture, This variety has a fast growth. So the farmers are using this variety to feed their livestock. So the second perennial variety, so this is the variety. Actually, it has basal non-elongating rhizomes. Also, it has the capability of regrowth across multiple seasons. That is one of the major character of this sorghum variety, as well as it has a grassy morphology, and the height is around 1.5 meters. And this is the third perennial variety. The height is also around 1.5 meters. It also has the capability of regrowth across multiple seasons. And but the main thing is here, it has more dense elongating rhizome system. This one also has grassy morphology, and we use these rhizome pieces to plant this variety. So statement of the problem. Now we know this forage sorghum is very much popular in southern Texas. But however, carbon sequestration and greenhouse gas emission dynamics studies remain poorly quantified. I found some research related to this. They have analyzed the forage yield and forage quality, also the drought resistant capacity. But I found only few articles related to the above ground and biomass amount and the soil carbon stock, considering the below ground and total ecosystem. Also, they didn't compare especially the seasonal carbon dioxide and seasonal methane emission data. So in this research, I'm going to cover these things. So the main objective of my research is to evaluate the carbon sequestration potential and greenhouse gas emission dynamics of annual and perennial sorghum cultivars under field condition in southern Texas. And my research, I have two specific objectives as well. The first one is to quantify the carbon sequestration potential of 3 sorghum cultivars based on soil organic carbon content and the above ground and below ground biomass content. And my second objective is to quantify and compare the GHG gas emission among these sorghum cultivars. In here, I am considering the fertilizer treatment as well. Also, as GHG gas emission, mainly I focus on methane and soil carbon dioxide emission. And my research, I have 4 research questions. The first one is how do these different sorghum cultivars influence on soil properties? So I focus on a few soil properties such as soil bulk density, soil total carbon, soil pH, and soil EC. Also, the second question is how do above ground and below ground biomass production vary among these 3 sorghum varieties? And the third question is which sorghum variety contribute more to plant carbon storage, total below ground carbon storage, and when we consider the total ecosystem carbon storage. And the final question is how do this GHG gas emission vary among these 3 sorghum cultivars? And significance of the study. This study provide the field-based evidence related to the carbon sequestration and greenhouse gas emission, considering these 3 sorghum varieties. So using the results, we can enhance the understanding about carbon sequestration dynamics of these sorghum varieties, and it will help to give the proper recommendation for the southern Texas as well. So ultimately, it will support the climate smart agriculture by increasing carbon sequestration potential and reducing the greenhouse gas emission. So the chapter 2, literature review. So literature review approach. In here, I have listed few sources I have used for my literature review. After I go through these sources, I could collected 120 research articles, and after go through the abstract and the paper, I sorted out to 80 research articles. So these are the number of studies used from the different region in the world. So next go to the conceptual framework. So in here, it's illustrate the different mechanisms involved in carbon sequestration and greenhouse gas emission in these 3 sorghum cultivars. So in here, in the same climatic and environmental condition, these 3 sorghum varieties going to input carbon to the soil, and it will alter the soil physical properties. It can be increased or decreased. Also, in the roots and rhizomes, the more carbon is going to store there too. So considering all of these, the below ground carbon storage is going to increase. And let's see what happened in the above ground carbon storage. So due to the carbon sequestration process, actually due to the photosynthesis process, the more carbon is going to fix through this above ground plant portion. And considering all the GHG gas emission also going to emit from the soil. So this is a simply conceptual framework of my research. So the chapter 3 methodology. Let's go to the study site. My study site is situated in Prairie View and Municipal Research Farm, and the annual precipitation is around 1,118 millimeters, and January is the coldest month, July is the hottest month here, and this soil type is Walkley fine sandy loam. And this is my research design. As you can remember, I have used 3 sorghum varieties. So each sorghum variety has 9 plots. So altogether we have 27 plots. So all these plots were arranged in our research field. Also here I want to mention one thing. We have used one treatment, the fertilizer. In here we have 3 fertilizer treatment as well. The recommended fertilizer, half recommended fertilizer, and control. And this all the recommended, all the fertilizer treatments were randomized within each of this variety. And this slide shows the dimension of my research plot. The length is 60 feet and the width is 7 feet. And we have 3 plant rows in one research plot. So before we go to the field, we did a germination test for the sugargrass ultra, that means our annual crop, and for the perennial seeded sorghum variety. So we selected 12 seeds from each variety and put in a petri dish and incubate in 28 degrees centigrade. So after a few days, we got the results. So if we go to the results, you can see sugargrass ultra showed the 100% of germination, but in this perennial seeded variety showed only 60%, and it took 13 days to germinate as well. So let's move to the management practices. Actually, this research were conducted over 2 seasons. In season 1, season 2. So season 1 is from June 2024 to November 2024. Season 1 from April 2025 to September 2025. So before we planting, we had to prepare our field. In the beginning of the season 1, we plowed all our plot and make the our land. But in season 2, we plowed only the annual plots only, because I'll tell you why. So this is the perennial plot you can see. So during winter, all the above ground biomass is died, but after the spring, at the start of the spring, so it's going to regrow. The perennials are going to regrow. So we didn't have to plow that field and prepare the field. So the management practices. So we used drip irrigation throughout the study period, as well as we applied fertilizer 2 weeks after planting. So we practice manual weeding at the first 3 months of the growing period, and in the season 2 we use 2,4-D herbicide for the annual and this prowl for the perennials to suppress the weed growth. And as you can remember, one of the major objective of my research is to analyze the carbon sequestration dynamics. For that one, I use 3 types of data: the soil data, plant data, and GHG gas emission data. And when we look at the soil data, We have collected soil data from two different depths, 0 to 15 and 15 to 45 centimeter in depth. We collected those data in three time period: before planting season 1, after harvesting season 1, and after harvesting season 2. And when we talk about the plant data, we collected above ground and below ground biomass data end of season 1 and season 2. And we also collected GHG gas emission data weekly throughout the season 1 and season 2. So let's move to the soil sample collection. So when we talk about the soil sampling, we use two types of data. We use soil intact core samples as well as the soil disturbed soil samples. Using these intact core samples, we measured the bulk density. And using these disturbed soil samples, we measured soil total carbon and soil pH and soil EC. After that, we got this bulk density and total carbon. We calculate the carbon stock in the soil. So soil analysis. So we calculate the bulk density. At first, we put our samples in the oven and dried in 105 degrees centigrade until get a constant weight. And after that, we use this equation to calculate the bulk density. And we get the soil total carbon using the CHNS analyzer. So now we have bulk density and total carbon percentage. So using those, we multiplied both of them with the soil depth, and then we got the soil carbon stock. So we measured pH and EC using these pH meters in our core lab. So the plant data. So as I mentioned before, we collected above ground and below ground biomass. So in here, we at end of each season, we selected 2 by 2 feet area and get our sample and oven dried in 60 centigrades in the oven until it get a constant weight, and we got the dried weight at the end. So using this dried weight, we could calculate the biomass carbon stock. And we need another parameter here too, that is above ground and below ground carbon percentage. For that one, we use the CHNS analyzer and calculate the carbon percentage of these rhizomes, roots, shoot and leaves. So now we have all these both parameters. Then now we can calculate the above ground biomass carbon stock using this equation. That is, we can multiply above ground dry weight by shoot percentage, and for the below ground carbon stock, we can multiply the below ground dry weight by the roots and rhizome carbon percentage. And after that, we can get the total biomass carbon stock by adding these two together. And then we calculate the system carbon stock as well. In here, I have two parameters. The first one is the total below ground carbon stock. For that one, I use soil carbon stock and the root and rhizome carbon stock. And the end, I calculate the total ecosystem carbon stock. So I use the same equation and I added above ground carbon stock in here. So finally, I measured the soil carbon dioxide and methane emission data. For that, I use the trace gas analyzer, and this is the soil collar. So we have installed this soil collar in all the 27 plots in my research field, and we mount this chamber on top of it, and we can get the soil carbon dioxide and soil methane emission data. And statistical analysis. So I use one-way ANOVA to compare the soil properties, carbon stock and the biomass data among varieties for each data sampling, as well as I use repeated measured ANOVA to analyze the soil carbon dioxide and methane emission, and I use JMP software to analyze this data. So now let's move to the research and discussion. At first, I'll explain about the soil properties. So let's move to the bulk density. So bulk density is a very important parameter because it gives the data about the soil compaction, and it is very important because it's give the root distribution pattern and the water infiltration patterns is directly influence on this bulk density. So as you can see here, I have illustrated the bulk density in two different depths, 0 to 15 centimeter depth and the 15 to 45 centimeter in depth here. So in here you can see the green color mean the annual sorghum variety, and this blue color represent the bulk density of perennial seeded sorghum variety, and this red color showed the bulk density of perennial rhizomatous sorghum variety. And without considering the crop variety, when we go to the deeper soil, we identify the bulk density is increasing. So as you can see, as example, I would say that in our soil, in the surface soil, the bulk density is between 1.3 to 1.7 grams per cubic meter, but in subsurface, it has increased 1.5 to 1.8 grams per cubic meter. Also, if we now look at the results, so if we look at the perennial varieties, the perennial variety showed a significantly lower bulk density than annual sorghum variety. As well as we saw the same observation in the deeper soil layer as well. In here, the rhizomatous sorghum variety showed a lower bulk density than annual and this perennial seeded sorghum variety. So if we look at the trend of this bulk density, we can see the annual variety. The annual variety means the green color line. It is continuously increasing its bulk density. But when we talk about the perennial varieties decreasing their bulk density. And in the subsurface soil layer also we saw that one. You can see the perennial rhizomatous sorghum variety gradually decreasing its bulk density here. So the main reason for that one is that perennial rhizomatous variety has more extensive rhizome, and this extensive root and rhizome system have improved the soil structure and the soil aggregation capacity. And let's move to the total carbon. So total carbon also a very important parameter here because it's directly influenced to the plant growth and the microbial activity. Here you can see in the deeper soil layer, this total soil carbon percentage is much lower than the surface layer. Actually, the main reason is in the surface layer, the microbial activity is much higher because all the residues and residues are going to incorporate in the surface layer, and the highest carbon storage also we can see in there due to the microbial activity. And but however, after 2 years of planting, we didn't see any significant difference in this total carbon storage here. And when we look at the trend of the total soil carbon, we can see the perennial sorghum variety, the perennial rhizomatous sorghum variety increasing its total carbon content. And when you look at the deeper soil layer, you can see the perennial seeded sorghum variety is the one who showing the high total carbon amount. And let's move to the total carbon stock. So as you can remember, I used this equation to calculate the total carbon stock, and there also we observed we didn't have any significant difference among these three varieties in terms of this total carbon stock. But however, here also we see that trend in this perennial rhizomatous sorghum variety, which is in this red color, increase the total carbon stock. In here also we observe the perennial seeded sorghum variety have increased the total carbon stock in there. So the soil pH is also one of the major important parameter. It gives us the idea of the alkalinity and the acidity of the soil. But in here also we didn't see any significant difference even after 2 years of planting, as well as in the EC, we didn't see any significant difference among these varieties. So now let's move to the plant biomass data. So in here I have illustrated the above ground and below ground biomass variation in season 1 and season 2. So if we look at the above ground dry weight, you can see the annual variety showed significantly higher amount than this perennial rhizomatous variety. Actually, we observe the same results in the season 2 as well. So in the season 2 above ground dry weight also, we got a significantly higher amount for the annual variety than these perennial varieties. And we observe the opposite in the below ground dry weight. In here you can see the perennial rhizomatous variety showed a significantly higher below ground dry weight than these two varieties, and same observation we observed in the season 2 as well. And then I combine the season 1 and season 2 data, and then I want to conclude what is happening to the above ground dry weight. So when we talk about the above ground dry weight, we can see the annual variety showed a significantly high above ground dry weight than other varieties. And the amount is the 26%, 26.4 megagrams per hectare. And we look at the below ground dry weight, we observe the perennial rhizomatous variety has the significantly higher amount, and it is 24.9 megagrams per hectare. And we want to look at some the literature data as well. In here we found a literature the saying at et al 2025, he has done this biomass evaluation for the perennial seeded variety, that means that blue color one. So he observed the perennial seeded variety produced 18.58 megagrams per hectare, but we got like nearly it is like 17.5 megagrams per hectare. And when we talk about the perennial seeded sorghum, perennial rhizomatous sorghum variety, the JSOP et al 2017 observed the above ground biomass of the perennial rhizomatous variety is 10.1, and we got it here as 11 points. So that we can say those numbers are matching. So then we look at the total plant carbon stock. In here we only observe, we consider this above ground part and the below ground, these roots and rhizomes. So before that, we calculate the carbon storage of this each above ground and below ground. So this is the table we got from the CHNS analyzer. And we look at the, if we look at the results, we can see the above ground biomass carbon stock is much higher in the annual variety, but the below ground biomass is much higher in this perennial variety. And when we look at the total carbon storage, we can see the annual variety is the one who showed the highest total carbon storage, especially due to this high above ground biomass carbon stock. And then we calculate the total below ground carbon stock as well. In here, we use these roots and rhizome data as well as the soil data. So you can see here, I have mentioned in here the soil carbon storage and the roots and rhizome carbon storage here. In here, we observe the perennial rhizomatous variety have the highest below ground carbon storage. Actually, this is one thing I want to highlight in this research, because if we are like talking about the carbon sequestration, we want to like keep it longer time in the soil. We have to keep, we have to store carbon in the soil, so we can use this perennial rhizomatous sorghum variety as a better variety for the carbon sequestration process, because in these rhizomes and soil stay longer time in the soil. And when we talk about the total ecosystem carbon storage, we have considered the above ground part and the below ground roots and rhizome part, as well as the this soil. So in here also we observed The annual variety shows the highest total ecosystem carbon storage because it has this higher above ground carbon storage. But perennial rhizomatous sorghum variety also got a considerable total ecosystem carbon storage here. And then let's move to the soil carbon dioxide and methane emission data. So in here, I have illustrated the soil carbon dioxide emission in season 1, soil carbon dioxide emission in season 2. In here, I didn't identify any significant difference among varieties, fertilizers, or any combination effect of variety of fertilizer. I do identified one thing, this called the control plots. The control plots emit less carbon dioxide to the soil than this fertilized plot, as well as the perennial rhizomatous sorghum variety showed a limited carbon dioxide emission than other varieties. And let's move to the soil methane emission. In here, one of the major observation is all the plots showed the negative methane emission. Actually, could be the reason for the methanotrophic bacteria in our soil. But however, we didn't see any significant difference among varieties or considering the fertilizer treatments in here. So let's move to the chapter 5, the conclusions and recommendations. So if we talk about the soil properties, the soil perennial rhizomatous sorghum variety significantly improved the soil properties. Actually, it is over time, the properties are improved. And when we talk about the above ground and biomass production, the annual variety produced the highest above ground production. So we can strongly recommend this annual variety for the ranching industry. And when we talk about the below ground biomass production, the perennial rhizomatous sorghum variety has the highest below ground biomass production. So we can say we can promote this variety for longer carbon storage variety. So we didn't see any significant difference among GHG gas emission among varieties, but however, we observed the higher carbon dioxide emission in annual variety mainly because due to the rapid litter turnover and the frequent disturbance of the soil. So these are some recommendations. Actually, when we talk about the carbon sequestration in soil, we can promote the perennial rhizomatous sorghum variety because it's gonna stay longer time in the soil. And another thing is, I would recommend a mixed system of annual and perennial rhizomatous variety because we can't remove the annual varieties in saying like it doesn't make higher below ground biomass production, but it produces the above ground biomass. So we can recommend this one for the ranching industry. And if we focus on the carbon sequestration potential, we can promote this perennial rhizomatous sorghum variety. And I would like to convey my deepest gratitudes to Professor Ramre for his continuous support, as well as my funding agency, USDA and IFA. Also, I would like to thank my committee members, all the members in our research group, the core lab, especially Dr. Sela, and finally my parents and my sisters and my friends for their continuous support. So these are some accomplishment I got during this 2 years period. So these are some publication I could complete during this period. So these are some international conference proceedings. So thank you very much. If you have any questions, I would love to answer it.
Okay, so thank you very much, Kusalika. Great presentation. Now the time for questions, concerns, comments, and suggestions. Anyone? Okay.
Thank you, Kusalika, for your excellent presentation. Congratulations. It was a great job. My question is on the selection of the varieties.
Yeah.
Are these the only varieties and why you select them? Second question, you have two depths
Yes.
range.
Mhm.
What's the base for those depths?
Okay,
Yeah.
thank you very much for the question. Actually, the first question is why did I select these three varieties, right? So, so we have selected annual variety and two perennial varieties. In there, we want to compare the annual and perennial varieties because the process is different. The amount of biomass, like the producing, the amount of biomass is different. So we want to compare the annual and perennial varieties and recommend we can recommend for the farmers, so what is the best sorghum variety they can produce, I mean they can plant in the soil in terms of biomass production or the carbon sequestration potential. And the second question is about the depth. So we have two depths, 0 to 15 and 15 to 45 centimeter. Actually we get the first depth. We want to, so our roots are like those are adventitious roots, so mostly the roots are in the surface layer. So we want to observe how the soil properties is going to change in the surface layer, as well as we use 15 to 45 because below roots also we want to observe how the soil properties is going to be changed. So that's why we selected those two depths.
Good. Yeah, but on top of that, the Texas A&M has developed the new two varieties, and those are the perennial one, right, Doctor? Right. So two perennials developed by Texas A&M, and that's not in commercial stage. I believe this is still on research stage, and this was submitted for the proposal to get to funding. So we need to have use the same varieties easily available because we can get the seeds from there. It's not available and commercially to purchase. So that was the main goal. So thank you for the question. Next. You can speak. Okay. No. I think somebody need to listen from the remote.
Thank you for your lovely presentation, Kusalika. So my question is, for your conclusion, you said utilizing the annual sorghum is the best one for ranching industry.
Yeah.
However, I noticed that throughout the different seasons, the bulk density tends to increase, especially for the annual sorghum. But amongst that, for the other two, the bulk density also increase. Now, how would you recommend to a farmer if they were supposed to utilize these species of sorghum for either zero grazing or grazing, right? As the bulk density increase, so does the porosity decreases, aeration in the soil decreases. What is the detrimental impact if a farmer was supposed to utilize these three species for their forage?
Okay, thank you very much for the question, Demar. Actually, yes, the bulk density is increasing if we plant the annual sorghum variety. But if you can see, if we plant the perennial sorghum varieties, the bulk density is decreasing. So if we manage to do a mixed cropping, I mean, if we can if we can grow both annual and perennial varieties together, we can have a balanced situation in there. As well as for the grazing also, it's going to increase the bulk density because of the animals. So if we use, so I recommend that the mixed cropping system.
How can they graze by 7, 8 feet tall? The plant comes up to 8 feet tall. Yeah, mostly.
Zero grazing, we're focusing on utilizing mechanical approach towards cutting that, so that increases the bulk density of the soil using machine.
Our understanding was they cut the forage and put it for the feeds later rather than go and grazing it. Yes, thank you. Next.
One of your questions, colleagues, I get inspired as an animal scientist. Kusalika, great work, and your team as well. I am curious, the effects of carbon capture through the plants on plant physiology and plant quality as a feed for animals.
is there a case for too much of carbon capture that would modulate the soil fertility, that would also modulate the plant quality as a feed? What are your thoughts on overexpression or carbon capture on the plant itself and on the soil?
Okay, so if we talk about the annual variety, actually the above ground biomass is much higher. So as for a farmer who is like in the ranching industry, I would highly recommend for this one, because the above ground biomass is higher, mean it's capturing high carbon dioxide as well, because it's all the gonna store in the this above ground biomass. So in the soil also, the soil, but when we talk about the annuals are gonna decrease. I mean, it's increase the bulk density. But if we plant the perennial varieties, so we can use perennial varieties to make like decrease the bulk density as well as to increase the soil organic carbon content. Yes.
So there's no negative effect of too much of carbon capture. for
Yes.
fertility.
Yes, sure.
Thank you.
Next question. You can answer the online question.
Okay. Do you think there are nutrient differences between varieties? Actually, I have analyzed these nutrients using the ICP in the core lab, but I could have time to get the results. So I think there is a possibility to have the nutrient differences. But before I say something, so I have to analyze the data. So yes.
Yeah, that's good response. Anyone else? I know this is a half day today and Thanksgiving holiday on the spot. So thanks for coming, your time and contribution. Oh, and happy Thanksgiving.
Before we close, I'd like to, first of all, great job, Kusalika, for your research. I think you did very well. Good example for other grad students. I just want to invite you on Monday. We have our Agro 5366 Principal of Environmental Science and Management graduate student term project presentations. Many of our students are here. They will be presenting their findings, which is about use of AI on emergent pollutant management. So it's a great work that they have been doing, and hopefully they will be publishing their work on peer-reviewed journals. So your attendance on that, it's 9 to 11, right before the other, there is another thesis presentation at 11:30. So before that, so I encourage you to attend if you are available. Thank you. Yeah, it's going to be in this room. Thank you for asking.
Presenting, major professor for the thesis.
There are 21 graduate students. It's the class I'm teaching.
Or before that, who's
No,
presenting?
after that 11:30, the thesis, that's
Oh, I see.
my student, the
Okay,
Bhagya.
very good.
See.
So I'm requesting you all to send an announcement to Dr. Pierre and Shidura is out today. But at the end, I want accountability. Our community needs to know about the wonderful learning opportunities. So please have them send out today and copy me, because I want calendar invite the entire college.
Sure, we already did that, but not the calendar invite yet.
Yeah,
But
so
we'll-
please send it, Dr. Pierre, copy me.
Sure.
Both of them, both the thesis and the seminars and the thesis, because this is priceless opportunity to learn from. So this particular graduate student taught us a lot. So learning is great, is that?
Yeah, and Kusalika is one of the presenters also, as far as Damar and others, so the students are
Excellent.
taking the
Thank you.
class, so.
Yeah, thesis invitation already gone in the schedule for all four of us, including her. All right, so
Thank you.
thank you very much for everybody. Happy Thanksgiving. What?
Another question too.
Oh, which question?
Another one.
Okay, go ahead.
Right. What do you realize by one-way ANOVA? I saw that you have your results for next two factors. Yes, actually, I want to say I didn't. I'm gonna answer this final question. So here, yes, we have collected the soil samples in 3 times before planting, after harvesting season 1 and after harvesting season 2. But in here, I didn't compare these 2 seasons. So that's that's why I didn't use two-way ANOVA, two-factor, like 2 factors. I didn't consider this one. So I only compare my varieties within that season. So that's why I used one-way ANOVA. I hope I answered the question.
All right. Thank you for any concern, comment, announcement, more? Good? All right. Thank you. Happy Thanksgiving for everybody. Take care.